Okay, so it's 4:30. I think we can start. Welcome everyone to our April afternoon in the science basement. Today we're going to have a really nice talk given by Sami Jalil about engineering humans, the future of gene editing. So I'm Flavia, I'm a member of the science basement, and before giving the floor to Sami, I want to just introduce our work in the science basement for everyone that doesn't know it. So the science basement is mostly a group of young scientists that really want to communicate science. We want to learn and we want to spread science all over. And we have a few projects to do that. This is the afternoon in the science basement, in which mostly once a month we host a speaker to talk about that job to the general public. In this way, we can learn how to communicate and people can get to know the science we've been doing inside the university. But we also have other projects like the podcast and science blogs where scientists write about their work and their lives as scientists and other projects as well. So if you want to learn more about these projects, you can go to our website, Facebook, Twitter, or Instagram page. This is the afternoon in the science basement team right now. And if you want to join our team or you want to do a talk like be a speaker like Sami today, feel free to message us, email us, and we'll be more than happy to welcome you in our basement. So, and also from this talk on in this spring session, we are now part of the program of the Year of Research-Based Knowledge. This is an initiative here in Finland to make research-based knowledge more visible and accessible to everyone. So they have several projects and joining their program this year is quite interesting. And if you are interested in more projects like this and watching more webinars and seeing more different programs, you can go take a look in their website and also their social media. Now, before giving the floor to Sami, we are going to put a quick poll just to get to know you people a little bit more, to know where our audience is coming from, how you're feeling. So now the poll is in. If you have like a couple of minutes to enter it.
All right. Hello, everyone. Good afternoon. My name is Sami Jalil. And here I am to talk with you about engineering humans, the future of gene editing. I'm really happy to be giving this talk, especially because I really want to know your opinion. It is a bit challenging to do this through Zoom, especially with the amount of people, so you can fill in the questions and answer section, and we will read them at the end, but we will try to make some discussion at the end of my talk anyways. Okay, so let's start. I was born and raised in Cordoba. Here are some pictures. Cordoba is a province in Argentina, and I love really the biodiversity there, and of course my friends and family, and I really miss them. But I traveled from Argentina to Finland to join Kirmos Bartiovara lab and starting studying gene editing. I am doing my PhD here. And as a side note, I really love Finnish nature. I love camping. I love watching the northern lights and walking on the frozen sea. Perhaps for some Finnish people, this is common, but for me, it's like living in a fairy tale. But let's start with the talk today. So the idea is that we have around 30 minutes of an introduction that I will explain. And in here, I want to give you the concepts that we need then for the debate. And after that, we will have these questions and answers, some polls that we prepared, and also the discussion. And I really look forward to this part. So, okay, I'm sure that you have seen or heard about CRISPR. It's all over the news. But how does the media talk about CRISPR? I bring you some examples here. This is the front page of Time, and they talked about CRISPR as a gene machine that will make experiments with humanity. Here we see this MIT front page with a Android-looking baby, and the message is how we can engineer the human race. Pretty scary also. Here in Nature, you see this Seek and Destroy, very, very strong titles always. And lastly, you can see here in The Economist, editing humanity, and you see a baby that now, thanks to this technology, it will have a perfect vision, perfect pitch. It will be a runner, high IQ, low Alzheimer, etc. They are selling this as a way of changing what we are as humans. But how much of all of this is true? And also, I would like to mention that last year in 2020, the Nobel Prize in Chemistry was awarded to these two ladies, Emmanuelle Charpentier and Jennifer Doudna, for creating CRISPR. So I hope I have convinced you that this is a big and important topic for this 21st century. But wait, let's rewind this story and come back to the basics. What is a DNA? What is a gene? And how we scientists can edit this. So I hope that you all remember these 4 letters from high school, AC, GT. These are molecules that are called nucleotides, and they attach to one another, forming the DNA. And these are the building blocks of the DNA. And a repetition of these nucleotides, so HECT, can be also called a sequence. You can think about this DNA as the recipe to create your own body. So as I told you, these nucleotides form the DNA, and in the DNA, we have our genes, and each gene contains the information to create a protein, and proteins are the substance that create our body. But for you to have an idea, the DNA, the whole DNA of one person has 3 billion of these nucleotides, and 3 billion is a huge number. It's so huge that we cannot really think about it. So I prepare an example for you. So imagine that after this talk, you go home and you find a book where your whole genome is written, and you start reading it at this pace, A, C, G, D. It will take you 95 years to follow your whole genome if you don't stop at all. So it is pretty big. But the amount of genes that we have is much smaller. We are talking about 25,000 genes. Anyways, as I told you, the amount of DNA is huge. And if you take into consideration that this amount of DNA is inside every cell of your body, and in one human body, we have approximately 30 trillion cells, this number is so difficult that we cannot really have an idea of how complex it is. And the DNA is what gives you the identity as a human, the identity as a Homo sapiens. This makes us humans and not, for example, a dog or a plant. But also, the DNA gives you the identity as a completely independent, completely individual person with a unique genome. So it is pretty important. And perhaps you also know that DNA is passed from the parents to the child. And this is how we keep the genetic information flowing. And as we discussed, the amount of information is huge, it's crazy big. So when we copy this information, we can have mistakes. And when a mistake causes a disease, we call it a pathogenic mutation because it is causing a genetic disease. And nowadays there are more than 4,000 diseases due to pathogenic mutations in one gene. And some examples that perhaps you have heard are sickle cell anemia, Huntington disease, hemophilia, muscular dystrophy. So if we can correct this small mistake in the DNA, we could save a person who has this kind of diseases. So the first technology that was created to tackle this problem was gene transfer. Gene transfer is an old technology. It is from the 90s, so it's as old as I am. And the idea here is pretty simple. So if you are a person who has a wrong version of a gene, a version that creates a disease, a scientist can create a virus, and it's not the coronavirus, we are talking about a completely different and safe virus here. A scientist can create a virus containing the corrected version of the gene that you are missing, and the virus can take this piece of gene and add it into your cells. So at the end, the patient will have two versions of the same gene, one version that is not working and one version that it is functional, it is working. And the disease of this person can be rescued. So this person can be cured with this technology. This may seem like science fiction to us, like designing a virus to transport a piece of gene into the cells. It looks quite science fiction, but let me tell you that nowadays in Europe, you can get some gene transfer therapies because it exists already in our reality. And here are all the products that are approved in Europe for gene transfer. These are all approved by the European Medicines Agency, so EMA. But as I told you before, this technology is old and it has some limitations because you cannot correct the gene that was broken in the person. You can just add another copy. So the newest version is this gene editing. So now imagine that the person has a gene that is not working because it has a mutation. We could have a tool that would scan or search in all your genome where is the mistake. And after it finds the mistake, it can cut it and replace it with a correct sequence. And then the person will have one version of the functional gene again. And this looks quite simple, but in real life, outside this PowerPoint, we are talking that the genome has 3 billion nucleotides, and we have at least 30 trillion cells in our body. So we are talking about a huge amount of information, and it's not so easy to do. However, there are clinical trials ongoing with this gene editing. There are more than 55 clinical trials. This means that more than 200 patients are being treated with these gene editing technologies, which is quite a lot. And we have 3 different ways of doing this, but the most popular one is CRISPR. And you can think about CRISPR about one single tool that can do 2 things. First, scan the whole genome to find the mistake, and then cut it out to correct it. That is what CRISPR does. But why then is this CRISPR so much popular than other versions of this gene editing? And it is because it's very easy to design, and we scientists love that. And then it is quite efficient. It can get the job done very fast and very clean. It is inexpensive. Creating these technologies, at least for research, it doesn't need a lot of money. It is quite reliable, and that makes it safe for therapies. And as I told you, nowadays we have more than 20 clinical trials going on with CRISPR. So there are real people nowadays being treated with CRISPR-Cas9. Okay, I hope that I have convinced you that CRISPR can be used to alter the human genome, to engineer our own genes. Now we should reflect about the applications. We could use CRISPR, for example, as a therapy, meaning that we can cure a disease that nowadays doesn't have any cure. But we could also use it for enhancement, meaning improving a human quality or even giving humans a quality that they don't have today. And also we can discuss about where to apply gene editing. We could apply gene editing in the somatic line, meaning that we can modify a particular tissue or a particular organ in our body with this technology. So for example, a person suffering from a genetic disease in the blood, we can correct the blood cells and then the person could be cured. But also this could be applied into the germline. And this means that we are not only correcting genes in the person, but also in all the children, great-grandchildren, and so on. So this mutation or this correction that we do in the genome will continue for generations. I think this is an important point, so we should discuss it a bit further. If you think about this discussion, it's something that 50 years ago, perhaps it was very interesting, but it was not very relevant because we didn't, as humans, we didn't have a way of doing this. So we didn't need an answer for this, really. But nowadays, we have the technology to do these changes in our genes. So as a society, we have to discuss what do we want to do with all this. So if you have a project where you want to apply gene editing as a therapy in the somatic line, you can think that you can save lives or improve the lives of people that have a genetic disease. And all the clinical trials that we have today, these more than 20 clinical trials in CRISPR, they are all focused on this part. And bioethically, there is not much to discuss here. For example, if you can cure a blind child, why wouldn't you do it? Well, this kind of technology may allow this kind of therapy. But we also have enhancement. And we could think that a scientist may do one person run faster or be stronger and be better at sports, be smarter, even live longer. So should we enhance human beings, giving humans new capacities that they don't have? Well, this is a difficult question. And if you think that this is all science fiction, let me tell you that this is already a thing in the Olympic Games, and the anti-doping international agency already has a guideline about how to detect and how to sanction genetic doping. So this is actually something that exists. Then we can think about germline gene editing therapy. So imagine that a couple both have a genetic disease and they don't want to have a child with this genetic disease. So basically, they could go for in vitro fertilization, adding here the sperm and the egg cells, and they could also add CRISPR to correct the mutation in the embryo. So at the end, they will have a new person, a baby, that has a correct version of the gene. This sounds okay, but you also have to consider that the genetic correction and all the possible genetic side effects of this therapy will be transmitted to all the children and great-grandchildren and so on. So in some, 1,000 years, you will have many humans actually suffering if something goes wrong in this process. So are we ready to change the human DNA in the germline? Well, this is also an unanswered question. And finally, we can use our imagination to think about a very science fiction scenario where a scientist decides to enhance humans in the germline, meaning that this new capacity will be passed through generations. So what kind of superpowers would we give to the future humans? will this be equal, or will this create inequality between edited humans and non-edited humans? Are we turning humanity into something else? Are we changing our species if we do that? What are the unpredictable side effects? So there are too many questions, and that's why there is a scientific moratorium in this germline editing, because we know as scientists that we have too many questions that are unanswered to start playing around with the germline. Or that was what we believed in 2018, because that year, Chinese scientist came out into a conference, and he announced that he did a genetic enhancement of 2 babies in the germline. He tried to make 2 girls called Lulu and Nana HIV resistant, so then they wouldn't get HIV if they were in presence of the virus. So this is a superpower that he chose to give them, and as we discussed, this will be transferred to generations. Here you can see that it was an international scandal in Yle, the Finnish news. There are many articles about this, actually. And what is the end of this story? Like, what happened with these babies? Well, we know that this scientist was sentenced to prison for 3 years. We also know that there were actually 3 genetic engineered babies, not just 2. We also know that the families didn't know that the babies would be genetically altered, so they didn't consent this. We also know that the identity of these babies and all the medical records are classified as secret to protect their safety, so we cannot really know what happened with them. And we also know that these gene mutations that they added were not made in a clean way, so they do have a lot of genetic side effects that we are not aware about. And then later studies showed that this superpower, this mutation that these scientists added into the babies, actually doesn't prevent them to get HIV. And there are some studies saying that it may actually be negative for these 3 babies instead of positive. So it was something. And what can we learn about this situation as a society, as humanity? So if we as humans really decide to start altering our genome for us and for future generations, we should take this decision together. It should be a collective decision. And also, we should guarantee equality. Look at what is happening now, for example, with the COVID vaccination. Some countries have vaccinated more than 60%, and some countries less than 1%. Will this time the rich countries do genetic engineering in their citizens, and poor countries will remain just humble humans? Also, this decision should apply internationally. There should be an international consensus, because changing our genome shouldn't depend on political parties or country borders. Of course, we need transparency. This kind of project should be transparent to the public and not being something done in secret. We really should have a clear scientific support, meaning that if we want to make a mutation in the genome, we should really know that it will help humanity, and not, like in this case, we are not really sure about the consequences. And lastly, we really need a risk assessment. We need to consider the risk for this generation and also for future generations. But then how can we get involved as a society? How can we have a voice in these decisions? So I have some suggestions, but I hope that we can discuss them together, because it's not that I have all the answers. But I think it's important to support science communication, such this kind of projects where we we scientists speak to the people. We should also try to participate in university events, trying to connect with the people who are taking these decisions. We should try, for example, to follow scientific podcasts to form our opinion, try to read scientific news. In most cases, when we just read the newspaper or the regular news, the science behind them is not so strong, and they are more trying to create an impact. So we need to access to more scientific information. And I also think that we should bring these issues to political debate, because nowadays, for example, we know what a politician believes about immigration or about economy and so on. But do we know what the president or the prime minister or the ex-political party, or do we know what they believe about editing the germline or editing human beings? Do we know what they think about artificial intelligence or this kind of disruptive technologies? So this should be more debated in public. And I strongly believe that publicly funded science should actually include us all and not just people inside academia. So I will leave you as a way of accessing to science information, 2 Netflix documentaries. And the good thing about them is that, for example, in this Unnatural Selection, they really have interviews with the people that are important in this story. Meaning, you will find, for example, in Unnatural Selection, an interview with Jennifer Doudna, who was one of the creators of CRISPR. You will see interviews with the CEOs of the companies that are selling these genetic and gene editing products. There are interviews with biohackers, who are people who are trying to change their own genes at home with homemade stuff. You will also hear about interviews with patients that are suffering from genetic diseases, and it's very important to see also their views. And in this other series, Explain, there is one chapter about designer DNA, one episode that I also highly recommend. So if you want to start forming your opinion about these technologies, this is a good way of starting. There are a lot of books also and scientific podcasts. But that's it for me. I will thank you. This is a picture of some of our lab members last summer. You can follow me in Twitter if you want to ask any other question, or just be updated about scientific information. And I also have to thank to the funding agencies that are supporting our research. And that's it for me now.